you want to make additional comments?
I figured there'd be questions.
Okay. Does anyone have any questions out? Any questions in the question box? Yes, Michael.
Yeah, thanks, Tom. Very nice work. I was wondering, we in the past have observed a difference in the biological activity of intracellular E2 versus secreted E2 in terms of binding to CD81. We've seen a very large difference with the intracellular form being much more binds with much higher affinity than the secreted form. I was wondering if you've looked at your secreted E1, E2 and compared the binding affinity to CD81 with the intracellular E1, E2.
Yeah, the binding affinities are pretty much the same between secreted E2 and secreted E1, E2.
I see. Thank you.
So I had a question which really is, for Tom or for anybody else. And then that is really the use of these animal models to test your vaccines. Do you expect, for example, if you use mice or guinea pigs or macaques, that you will get different results, that they will each corroborate the other? What is your thinking on that?
Well, I mean, we're looking at the quality of the immune response right now. In the mice, we immunized, for instance, with 70 micrograms. In the macaques, we used 100 micrograms as a priming dose. And there's a large difference in the size of the animals, of course. And so what we're looking at right now is really the overall magnitude of the endpoint titers we see, and compare that to the quality of the neutralizing antibody responses that we see. I mean, we have no way of knowing whether the mouse would translate into what we see in the macaques and the humans. What we can say right now in our initial macaque study is that the overall titers are lower than what we see in the mouse system. But we do see a higher endpoint titers using membrane-bound E1E2 when tested against membrane-bound E1E2 down on the plates compared to secreted E1E2. But again, when secreted E1E2 is on the plate, they're about the same. But we see higher neutralization against homologous isolate using the membrane-bound E1E2 as a vaccine. But that differential seems to taper off when we look at heterologous neutralization titers. And so we're just trying to sort of get our hands around the serological responses at the time, and then improving the valency, if you will, of secreted E1E2 in a nanoparticle approach, so that it would be more comparable to membrane-bound E1E2.
Jens, do you have any comments on this particular question? I mean, you're still working with chimps in Denmark, are you?
Chimpanzees in Denmark, yeah. No, I don't have any particular comments on that, right?
I might add, Roger, if you have a few more minutes, that we can make these things that we call virus mimicking polymer assemblies using the polyprocessing adjuvant and delivery system. And as such, we go ahead and decorate the surface with E1E2 and encapsulate the core protein as an example. And we can do this because of the ionic differences between the two antigens. And at least in mice, we can show that we can stimulate CD4 and CD8 responses to both E1E2 as well as to core. And so I think this is one of a novel approach using the polyprocessing-based system to make these what we call virus mimicking polymer assemblies that mimic a core, HCV, E1 and E2 plus core, or it could contain NF3 or T cell epitopes, if you will. as a delivery vehicle. So I think it's rather novel in that regard. And so we've had promising results in mice.
Michael?
Yeah, just a comment about the use of rhesus macaques. You know, I think any species is good if you're comparing different vaccine candidates to, you know, as an initial screen, whether it's mice or rhesus macaques. But I would like to caution about expecting a translocation from macaques to humans, a predictive effect in humans from macaques. And the reason I say that is we published many years ago a vaccine comprising the nuclear capsid protein of Hep C, made in bacteria, highly purified, adjuvanted with ischamatrix, a pretty powerful adjuvant. And in macaques, we saw incredible CD8 positive T cell responses that were incredibly long-lived. And so we were very excited. And then with Australian colleagues, we did a phase 1 trial in Australia. And we got good CD4 responses, but the CD8 responses were quite weak. So, you know, I kind of noticed that there's a lot of emphasis on using the macaque model for HIV vaccines. And, you know, while you would think they would be more predictive of human responses to mouse data, what I'm saying is that that doesn't necessarily come out that way. So I think you don't know about a vaccine's immunogenicity until you test it in humans. So I would be cautious about having a screen in the macaque.
So what would you say is the best reason to test it in macaques?
Well, I mean, personally, we have our own strategy for eliciting cross-neutralizing antibodies and cross-reactive T cell responses, which I'll describe tomorrow. And my feeling is I just want to go into humans rapidly. I think going into macaques is a delay, and it's not necessarily predictive of human responses. So you could miss a good vaccine by testing it in macaques and dumping it.
Thanks, Michael. Any other comments from any of the others?
This is Manson Law. So we did done some macaque work, and surprisingly, antibody response actually pretty interesting. And I'm going to talk a bit about that more as well. But in terms of the T cell response, we didn't measure, so we wouldn't know, right? But in terms of neutralizing antibody response, we went in all the way, like from neutralization, epitope mapping. All in all, the response is quite interesting, very similar to the human studies. And you did and Sharon Flay and did before, right? And surprisingly, when we go down to look at the monoclonal antibodies level, we found some of the antibodies actually look like the human body neutralizing antibodies, which use the VH169 gene. In the macaque, it actually use a VH1.36, which is the closest VH169 genes in monkeys to make the body neutralizing antibodies bind to the same place for cross neutralization. So it was a pretty interesting model. I wish I know more about that. But so far, in terms of antibody response, responding to E1, E2 immunization is quite similar to what we see in human at that level. Yeah.
There's a question from Dr. Rama Amara for Dr. First. So in non-human primates, how many doses are required to induce neutralizing antibody response?
Right, okay. We're doing the serology right now, but at least a prime and 2 doses. We're doing the kinetic analysis at the present time, so I can't tell you any more than that right now.
So I don't see any more questions. Maybe
There are a few
we
in
can
chat,
have
Rajan.
Sorry?
There are a few questions in the chat.
Oh, really? I
Yeah.
didn't see any in the
Yes.
chat. No open questions. those questions were for previous speakers. There's nothing in the chat that I can see.
Oh, I think there's a question from John Law, which I answered in the type, which would be good to air. It was about the animal models. John said many models show immunogenicity to vaccine candidates, but none show the ability to reduce virus chronicity, which is the key clinical outcome in which we're interested in. And I would agree with that. I mean, I think the animal models have they all show limitations currently, including the chimeric liver and the humanized mouse for the assessment of vaccine candidates.
There's a question I see here from Mattia Bonsignori. Can you comment on the sequential versus cocktail approach? Is that for Dr. First? Doesn't say. Yeah, it should be for him.
Oh, okay, so
Yeah, it is.
this is actually an open question regarding, an E1E2 consensus design. Now, let's say a common to all of the 6 major genotypes, and we've done work versus, let's say, a cons genotype 1 or a cons genotype 2 or a cons genotype 3. And whether it would be worthwhile moving forward with a consensus sequence all, in which when we design this, we can show a significant improvement of binding to the E1E2 complex antibodies by about 5 to 10-fold when we make cons all sequence, and use that as the primary E1E2 vaccine candidate going forward. It could be with or without or NS3 or peptides, or whether it be more of a cocktail approach. But let's say a cons 1, a cons 2, and a cons 3 as a multivalent vaccine. And so we've done work with cons all, and we've made cons 1, and we're doing cons 2 and 3 right now. But this is an open question, of the overall formulation, if you will. Cons all versus focus on consensus sequence, for instance, for genotype 1, and then genotype 3, and then use that as a formulation. So we've done in that area. We've showed that we can significantly improve the antigenicity using a rather unique approach. But this is just sort of an open question there for all the panelists, what they think about that approach.
David Thomas asked the question, is there any basis for some envelope epitopes functioning as decoys to divert attention from epitopes that are more lethal to the virus?
David Thomas.
Sorry.
Is that question directed to?
I think
I guess that could go to
directed
Arash,
it to you.
or Justin or Andrea,
Yeah.
anyone, any of the immunologists. I don't know. It may be a bad question. I was just curious about, valuing them all equally in the.
Yeah, and David, that's a great question. I mean, the truth of it is, I probably don't know. We don't know much about E1 at this point. A lot of work has been concentrated on E2. A lot of subjects who have antibodies to E2, they may have E1, but you usually don't find anybody that has antibodies to E1 but not to E2. I was misguided myself, thinking that HVR1 was, presumably something that, was there to target the antibodies, but I think the recent data with Rob Honegger and others, that may not be the case. Usually, I mean, that's something that's really interesting that you bring up, because in a lot of viruses, like hepatitis E virus has this soluble ORF2 protein, just like maybe flu and yellow fever. So sometimes, people think that these soluble proteins are put out there to divert your immune system. On a different note, we think that it's actually, may not be the case for every virus in that way. That's not just sort of, diverting your antibody response. They have actually, particular roles. But that being said, with Hep C doesn't really put these soluble proteins. And so, yeah, I'm not sure that they're there to necessarily divert, but what is interesting, and I think this has been, at least from my talking to many other investigators, that, certainly in the rat model or something, is E2 doesn't seem to induce much of an antibody response. Now, whether that's because your immune system thinks, over the years and evolution, whether this protein has adopted a sort of a response that says your immune system thinks it's a self protein, and therefore it shouldn't mount a response to it. Maybe that's a sort of a possibility, but it's a long-winded answer of saying I don't really know much at this point.
There's a question from Paul Gipford. I hope I'm pronouncing correctly. How do you choose which HCV strain for challenge?
Well, this is an area that Alex Kloss would be engaged with, but that's a great question. It's going from homologous challenge to the more diverse genotypes, and so it would be done in a sequential fashion: genotype 1, genotype 2, genotype 3.
There's a question from Anna Chumbe. Sorry, go ahead.
Sorry, I wanted to go
Go.
back to the question that David Thomas raised about HVR1 immunologic decoy. I think that issue has been tackled. There's no doubt, all of us know that HVR1 is highly immunogenic. And in fact, it's immunodominant. You pretty much see it in virtually everyone. And there's been quite a number of studies to see if that region can be dampened by deletion. The problem is that when you delete the HVR1 region, what you have is an E2 that's even less immunogenic. So there are other factors involved or other roles of HVR1 that makes it pretty important, in that if the overall immunogenicity of your E2 molecule is drastically reduced without HVR1, then it may be that HVR1 is still required. So it is no doubt that it's easily escaped, as we all know.
I think Jens made a mutant, a construct that was absent HVR1, if I remember, and inoculated it into animals.
Yeah, we did, and of course it's a long story with the HVR1, and there's many aspects to that, but sort of from the life cycle point of view, there's no doubt that the HVR1 also had a role there. The animal was negative, literally, or was barely positive for weeks, and then it adapted and got some E2 mutations that kind of compensated for the lack of HVR1. I'm not sure we ever understood what they did, but it's clear that the virus can easily persist then without HVR1, and it's also clear in cultures later that we and others, Thomas Speechman and others, showed that you can take it out of culture viruses, and you can get viruses that are quite functional, at least for most strains. Yeah. And then, of course, I guess that's what Stephen was getting at, is what is HVR1 then doing? And there's no doubt that HVR1 is immunodominant. If you look for antibodies specifically towards HVR1, they're going to be there in many cases, and maybe you lose them over time because they don't recognize whatever you're using to test with. But so then of course but then it has all these other functions, and of course many people have looked at that too. And that's when it gets interesting. I think that it clearly has a role in shielding, in some ways, other epitopes, but it also interacts with other sequences in E2, and it sort of in a way determines the overall neutralization of the virus in very complex ways. And I think we still have more work to do to understand that.
I should have linked in the
Yeah.
question back, those very first persons who inject drugs who either persisted or cleared that I showed in my talk, Stuart Ray looked at the virus sequences, and one of the striking differences was the accumulation of non-synonymous mutations in those who ultimately, in the HVR1 and those that persisted, suggesting that they acted like a decoy. I can't say anything smart about that. Maybe I saw Justin pop his picture on, appreciated Stephen's comment.
Heidi Drummer has a construct in Melbourne. She's been working on for a long time with all the hypervariable regions deleted from E2, and we've expressed that in an adenoviral vectored construct alongside a T-cell immunogen recently, and she's done the neutralizing antibody work, but it looks reasonably immunogenic. So I mean, the function of HBR1 and removing that for use in a vaccine immunogen are kind of two different issues, aren't they? I would say.
Yeah,
But I think it's a strategy to be explored for sure. We should be exploring the strategy of removing these hypervariable regions where there's such good evidence that the human antibody response can just escape from these regions so readily. I mean, it ought to be a sensible strategy to look at, I think.
So we'll have one last question from Anna Chumbe. Tom, what do you think should be the effect of the membrane part in the membrane-bound E1/E2 vaccine?
I mean, that's a good question. I'm not sure if there have been T-cell epitopes mapped to the transmembrane regions or not. I don't know. What I can tell you is without the transmembrane regions in a secreted form, we have a better defined vaccine. It's a more uniform molecule. And so perhaps someone else can address really what the potential benefit of the transmembrane region would be on E1 and on E2 in terms of any potential immunological response to that region.
Yeah, so I guess what you're saying really is because the removal of that domain facilitates purification, expression, and purification, and makes it a more homogeneous product.
Absolutely.
That's the non-immunological response.
That's correct.
So it might be worth thinking about that. So I think we can give it back to John now to
Okay.
close us out.
Good deal. Thank you all. That was a great discussion. We really were getting a very interesting perspectives on ways forward there over the last 15, 20 minutes. I greatly enjoyed that. We have an evaluation poll. Please fill those out. I quickly want to thank Dave for kicking us off and really setting the stage for why we still need a hepatitis C vaccine to achieve goals for hepatitis elimination, the biologic circumstances that make it feasible as well as challenging. Andrea talking about the lessons learned from the previous vaccine trials and some of the differences between immune responses that was continued by Arash in his look at the T-cell versus humoral immune responses, and then Tom looking at rational vaccine design. We're going to continue this tomorrow. Perhaps we can show the agenda for tomorrow. We'll be meeting again at noon. And you see the list of speakers there, including Michael Houghton, who we've already heard from today, and then we'll have a panel discussion to end the afternoon tomorrow. And again, all of this will be on our website at globalhep.org to view all of this material for the future. So please spread the word to people who are unable to attend today. I think we have we do have some upcoming NIH elimination, hepatitis elimination webinars scheduled one by National Institute of Drug Abuse on May 5th, and one from the National Institute of Minority Health and Health Disparities on the 16th of June. We'll be giving you more information about that as those agenda are developed. I think we also have one other advertisement for another symposium coming up with related to hepatitis C virus, scheduled later in July with the information shown here. So I want to thank again Raj and Jake for moderating today. Thanks again for all of the speakers. Please come back and join us tomorrow as we continue this discussion regarding development of hepatitis C vaccine. Thank you. Thanks again.
Thank you all.
Well done, guys. Very good. I really enjoyed it. Thank you.